The last speaker is Shiu-Feng Huang. He got a MD degree on 1983 and got a PhD degree on 1997 in Institute of Pathology and National Taiwan University. Now he's a full professor and investigator and attending physician in the Institute of the Molecular and Genomic Medicine, NHRI. Today he will introduce Taiwan Liver Cancer Network to us. Professor Huang, please.
Thank you. It's my great honor to come to give a talk introducing Taiwan Liver Cancer Network. And I just heard the president saying about the collaboration and including the research. I think our network can have some contribution. Taiwan Liver Cancer Network was established since 2005, all supported by government's fund. The background, as you know, for the many biomarker study for the new drug, biobank is actually very important, a good quality biobank. HCC is the major liver cancer and major cause of cancer death in Taiwan for the past 20 or more years. So our government picked up this is a major disease. And our specific aim is to collect the liver. Initially, it's called liver cancer. Actually, we recruit all patients with liver tumor. If we can get a specimen, as long as they agree, we were including. So actually, we include most patients come with the liver disease. And we also want to collect the clinical data and the survival data, which is very useful for our applicant. And finally, we want to establish a research network to provide sufficient clinical and survival and specimen and a good quality specimen. Our design is we collaborate with several big medical centers, and we're following a standard protocol. So everything inside the collecting protocol, the clinical data is standard. And we build a research team in each medical center with well-trained research nurse provided everything, because the doctor is all very busy, so this station nurse is very important. And our specimen is centralized collect in the NHI biobank at Zhunan, and we all store in the -80. We didn't use the liquid nitrogen. This is more easy to handle. And you can see our distributions that we have 2 medical centers north, 1 in the central and 2 in the south. So this is our website. It's quite easy. You know, as long as you type Taiwan Liver Cancer Network, you can check our application list, how to connect. These are the 5 medical centers picture. You can check them, and it's very easy. And our current progress by the end of last year, we already recruit more than 9,000 patients and have near 5,000 patients have frozen specimen. Near 8,000 have blood sample, and we also have collect our paraffin block. So our specimen is quite complete and quite big amount. And so far, we already have 147 applicants, and they can apply like blood DNA, serum, plasma, tumor DNA, RNA, protein, tissue, and our paraffin section, tissue array section. So far, we already sent out more than 67,000 specimens by the end of February. The distribution of this, our case, you see we have specially analyzed about the cholangiocarcinoma. Majority, more than 80% were HCC, but we also have cholangiocarcinoma of 400 patients. And you see they are increasing combined HCC and cholangiocarcinoma. And among them, we have many have frozen specimen. Majority still HCC, but we have this 200 cholangiocarcinoma and combined. Actually, even though it doesn't like a big number, but this tissue is frozen tissue. Actually, is quite precious because many cholangiocarcinoma is inoperable. It's impossible to collect a large amount of specimen. But we even have cholangiocarcinoma tissue array, and this is not just tissue array. They combine with very incomplete clinical data. Also have combined the DNA, RNA, even the blood sample for genetic study. So our niche is the TLCN is the collect is very representative. For Taiwan, because they cover from north, middle of South Taiwan, and they will have a central pathology core lab to ensure good quality and correct diagnosis. And we have complete follow-up data. And we have this tissue is in charge by the liver cancer. Liver cancer is the central pathology core. So in addition to good processing, storage, and good quality control, we need the DNA, RNA need to be good quality before they can send out to our applicant. And accurate diagnosis, this is important part because you see this is our paraffin section. Most of the pathology department won't send out their paraffin block. So when we recruit patient, collect specimen, we prospectively already recruit our own paraffin block, and paraffin can cut section, can do many IC staining. Nowadays, can even for the genetic diagnosis for NGS is all. We are very fortunate. And one thing special for this biobank is that because I'm a pathology, everything tissue, when the frozen tissue, when they send for DNA or RNA extraction, we need to do a frozen section to double check the quality. Because most scientists, when they get a sample from the doctor, clinical doctor, they didn't check it. So sometimes they didn't know what is inside the frozen section. It's just according to the pathology report, but it doesn't equal. For this one, it's all HCC. But for this one, you can see a lot of fibrous tissue, the tumor gland. This is usually it's cholangiocarcinoma. They have low severity, so their DNA may be a low amount. So we need to let the applicant know the content. And some, you see the big tumor, actually they all necrosis. This is common in for the HCC. Status post TAC, then most are necrotic, and this kind of tissue is not good for research. And some have scarring, so even though they look wide and firm, actually there's no tumor. All these kind of specimens won't be sent out for our applicant, but their clinical data and the blood sample will still can be used. In Taiwan, HCC is our major cancer, and 80% may have a viral infection. So we prospectively collect all cases. As long as they have blood sample, we will send for HBV DNA check and HCV RNA check if they were positive for anti-C antibody. For patients, even their HBs antigen is negative, we still will check for the DNA, because some patients, even they were negative for antigen, they will still be positive for HBV DNA. So we identify quite a lot of patients, maybe around 10% of patients with occult HBV infection. You can see we have the copy number data, genomic, genome type data. So our patient, when our applicant come to apply for the clinical data, we will include this accurate viral data. And actually, many cholangiocarcinoma in Taiwan also have chronic viral infections, so this data is quite useful. And the second niche that we all our biosample collect in one place in the central part, because many hospitals they build a consortium of biobank. Actually, the sample is in different hospitals. So their data and the sample are not centralized, and sometimes it's difficult for application. And we have a working committee of the scientists. They were neutral and they were very devoted, and they were not. Actually, most of them not come from these collaborating medical centers. So they will try to be neutral and try to be friendly to our applicants. So our biobank is well known for its neutral and friendly for our application. Our applicant can come from every place from Taiwan. This is our tissue array. We have cholangiocarcinoma tissue array, have HCC tissue array. They were very popular for application. And this is a typical case for cholangiocarcinoma because the tumor gland usually have a very fibrotic. The tumor presence is quite low, and this is actually not good for DNA extraction. Usually we like to have high tumor component. But so to solve this problem, we have, get support from our government, from our institute to get a laser capture microdissection, so that for these kind of, usually we do that for the cholangiocarcinoma because the case number is small and the DNA is precious, but we have a lot of HCC, so if this tumor person is low, we would choose another case. And you can see the quality, this DNA quality is quite good. It's a major band. since we have website, we often see the request for emails that can they apply, over other country. So far, because Taiwan liver cancer center is supported by government, we cannot accept application from scientists from other country or foreigners directly, and so need to be Taiwan scientists or domestic industry. But by collaboration with Taiwan scientists, you can apply for that. So when the Cholangiocarcinoma Foundation, if you can introduce that to, come to collaborate with our scientists here, and we can do that study. And we have a lot of application and come from all places in Taiwan, or other major medical center or research institute. We even have 3 industry applications. Now we are promoting that industry can direct to apply our cases. Our application are customized. They can apply whatever they want. So you can see this is some example. Now some would like to have early or late recurrence, some like a different stage, HBV, HCV, like that, and some would like to have like blood sample for GWAS, 1,000 case, because we have like 8,000 blood sample, and some would like to mail HBV and a high copy number, and some would like paired RNA tissue fragment for like HBV, HCV, and non-B, non-C. We can all met their request. And we also have several, but fewer, much fewer cholangiocarcinoma applications, such as because the tumor, for RNA extraction, maybe the stroma also plays some role. So they will apply for like tumor predominant and stroma predominant, mean the low tumor percentage for the pair RNA or paraffin section. And also some apply for tissue array for cholangiohepatoma, and some for the RNA, and some for the non-B, non-C. And this is another example for like, okay. And so far, our Taiwan liver cancer center have very good publication from our applicants. So far, already 74 publications, and among them, many of them were into very high impact journal like hepatology, gastroenterology. And finally, I would like to say that because this is a teamwork and collaborate with the 5 medical center and the research team in NHRI, so we like to acknowledge all our team worker in each hospital. And thank you very much.